Hey everyone, in this lesson we are going to talk about glycolysis. So glycolysis, lysis means breakdown and glyco here indicates glucose. So basically glycolysis is breakdown of glucose, and this glucose can be supplied from any sugar, and this could be dietary sugar, fructose, galactose or glycogen, as well as it can also be supplied from any catalytic reaction, catabolic reaction. So suppose when glycogen is broken down into glucose, so that is one example of catabolic reaction where glucose is supplied, and that can be again used to synthesize energy. Now the other thing you need to know is there are some cells or tissue which heavily relies on glycolysis to synthesize energy in the form of ATP. So some tissues such as brain, kidney, exercising muscle and poorly vascularized tissue such as cornea, so uh, these tissues are heavily rely on glycolysis to synthesize energy. Now, the example for cells such as red blood cells, which lack mitochondria, and sperm cells, they also rely heavily on glycolysis to synthesize energy. As well as the cells which have mitochondria, they can also use glucose to synthesize energy in addition to the other substrates such as fatty acid, proteins, or amino acids. So now when glucose is broken down, it synthesizes two molecules of pyruvate. That means from one glucose, two molecules of pyruvate are synthesized, and in this there are total 10 reactions of glycolysis. So in the first phase, which includes reaction 1 to 3, it's called energy investment phase. It is energy investment because in this reaction, two molecules of ATPs are consumed during this reaction. And the second phase, which is reaction 4 and 5, which is called a splitting phase, because in this phase, one molecule is splitting into two different molecules. And in the third phase, which are reaction 6 to 10, which is also called as energy generation phase, because in this phase there are four molecules of ATPs are synthesized as well as two NADH coenzymes are synthesized. So now you can see that one molecule is broken down into two molecules of pyruvate, and this reaction can take place this involves 10 different reactions in three different phases. Now this splitting phase is optional. If you don't want to remember, this can be included in the energy investment phase, and this is what you usually found in most of the textbooks, but for me, I find this is easier way to remember, having including one more phase. So it's up to you how you want to remember. It doesn't really matter. Now from glycolysis, so as you can see here that the first phase where two molecules of ATPs are consumed, and the last phase involves synthesizing four molecules of ATPs. So together there is a net gain of two ATP molecules. So therefore glycolysis yield two molecules of ATP. So one glucose yields two molecules of ATP. And also remember that glycolysis takes place in the cytoplasm. So now let's talk about how this glucose is transported into the cells. The thing is, glucose just cannot directly diffuse into the cell, but it can be entered by one of the two transport mechanisms. First is sodium independent facilitated diffusion transport. So in this transport, it involves transporter, so there are which are called glucose transporter. So there are 14 different glucose transporter in the cells, so which are also called as GLUTs. So there are different 1 to 14 glucose transporter. So what happens that when glucose actually binds to this glucose transporters, it results in the conformational change in the glucose transporter, and that will eventually transport glucose molecules from extracellular space into the cytosol. And because this is a facilitated diffusion transport, glucose movement occurs down to the concentration gradient, meaning glucose is transported from the area of high glucose concentration to the area of low glucose concentration. Therefore glucose movement occurs down the concentration gradient. Now, the other thing you need to remember that the glucose transporters display a tissue-specific pattern of expression. So, for example, glucose 1 is abundantly present in or expressed in red blood cells, blood-brain barrier, as well as it is expressed in low amount in skeletal muscle. Now, glucose 2 is expressed in liver, kidney, pancreatic beta cells, and they are also insulin independent. Now, whether they are insulin independent or dependent, I'm not going to talk about this in detail, and I will definitely have a separate video on this glucose transporter where I'll be talking about this in detail. Now, GLUT3 are expressed in neurons. GLUT4 transporters are expressed in adipose tissue and skeletal muscle, and these transporters are insulin dependent. And GLUT5 actually, opposed to glucose, it actually transports fructose in small intestine and testes. Now, the second transporter system is sodium dependent co-transporter system. So in this system, glucose is transported against a concentration gradient, meaning glucose is transported from an area of low glucose concentration to an area of high glucose concentration. And it is also called as co-transporter system because two ions of sodium are co-transported along with glucose molecule. So therefore it is called a sodium dependent co-transporter system, and the transporter, which is called as sodium glucose dependent transporter, also known as SGLT, which are responsible for transporting glucose in this manner, and they are heavily expressed in intestine, renal tubules, and choroid plexus. So now let's learn the step-by-step reactions of glycolysis. So first consider that now glucose is now transported from the extracellular space into the cytosol. So once inside the cytosol, so suppose here we have a glucose molecule, and I have also labeled the carbon atoms in the glucose molecule. So once inside the cytosol, glucose is first phosphorylated into glucose 6-phosphate. And and the enzyme which is responsible for this reaction is glucokinase or hexokinase. Now, these enzymes are expressed differently in different tissues. For instance, hexokinase 1, 2, and 3, they are expressed in every metabolizing cells in the body except for liver and pancreas. Whereas glucokinase, which is also called as hexokinase D or hexokinase 4, this enzyme is only expressed in liver and pancreas. Now, another important characteristic of kinase enzymes are these enzymes actually they transfer the phosphate group to other molecule. So in this case, the phosphate molecule is obtained from ATP, and this phosphate molecule from ATP is transferred into the carbon 6 position of glucose to synthesize glucose 6-phosphate. So remember that any kinase enzymes, whenever there is an involvement of kinase enzyme, what they do is they generally transfer the phosphate group to the other molecule. Now also remember that this is first irreversible reaction of glycolysis that consumes ATP, and also this glucokinase and hexokinase enzymes, they are heavily regulated, and I'm not going to talk about the regulation of this enzyme in this video, but I will definitely have a lesson on regulation of glycolysis in the next video. Now, what is the significance of this reaction? Why is this reaction so important? So the reason behind that is phosphorylation creates a negative charge on the sugar molecule, and plasma membrane is essentially impermeable to glucose 6-phosphate. Furthermore, there are no specific transmembrane carriers which carry this compound across the membrane because these compounds are too polar to diffuse through the lipid core of the membrane. Therefore, the irreversible phosphorylation of glucose effectively traps the sugar as glucose 6-phosphate in the cytosol, thus committing it to further metabolism in the cell, that meaning that it is further committing to the subsequent steps of glycolysis because it cannot transfer it back into the extracellular space. Moreover, this rapid conversion of glucose to glucose 6 phosphate also keeps the intracellular concentration of glucose low, so it kind of regulates the intracellular glucose concentration. So this is the first step, and it is very important step and highly regulated step. Now next step is the isomerization of glucose 6 phosphate. So glucose 6 phosphate is isomerized into fructose 6 phosphate, and the reaction is catalyzed by phosphoglucose isomerase. And in this reaction, the carbonyl oxygen of glucose 6 phosphate is shifted from carbon 1 to carbon 2 position of fructose 6 phosphate. And as you can see here that glucose 6 phosphate and fructose 6 phosphate, they are isomers. So isomers are basically compounds that have the same chemical formula. So this both glucose and 6, they have the same chemical formula but have different structure. For example, if you see glucose and fructose, it is C6H12O6. So the same formula but have a different chemical structure. So, and then therefore the enzymes responsible for this reaction, they are called isomerase. So I'm just telling you because you don't need to memorize each and every enzyme. If you know these are isomers, you know that the enzyme responsible for this reaction would be isomerase. Now the third reaction is phosphorylation of fructose 6 phosphate. So fructose 6 phosphate is phosphorylated to fructose 1,6 bisphosphate, and the reaction is catalyzed by phosphofructokinase 1. And here you can, as the name suggests, because it is kinase, in this reaction the phosphate group is transferred from ATP into the carbon 1 position of fructose 6 phosphate to synthesize fructose 1,6 bisphosphate. And this is the second irreversible reaction that consumes ATP. And again, remember that this is also a very highly regulated enzyme, and I'm going to talk about this in the next video. Now the fourth reaction is cleavage of fructose 1,6 bisphosphate. So in this reaction, fructose 1,6 bisphosphate is cleaved between carbon 3 and carbon 4 position to synthesize triose phosphate. And in this triose phosphate are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. An enzyme responsible for this reaction is called aldolase, which is also called as fructose 1,6 bisphosphate aldolase. And this form of cleavage is called aldol cleavage, which is again catalyzed by aldolase enzyme. Now another thing you need to remember about this reaction is dihydroxyacetone phosphate cannot enter into the third phase of glycolysis, which is the energy generation phase. Only glyceraldehyde 3-phosphate can enter into the third phase of the glycolysis. Therefore, dihydroxyacetone phosphate must convert into glyceraldehyde 3-phosphate in order for glycolysis to continue. So therefore, our fifth reaction of glycolysis is isomerization of dihydroxyacetone phosphate. So this is the isomerization of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, and the reaction is catalyzed by triose phosphate isomerase. And as you can see here that both dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they are isomers of each other. So therefore the enzyme responsible for this reaction is also called isomerase. And by doing that, we also end up having 2 molecules of glyceraldehyde 3-phosphate and at the end of splitting phase. So remember that from one glucose molecule, now we have 2 molecules of glyceraldehyde 3-phosphate, which is now ready to enter into the third phase of glycolysis. So next reaction is the oxidation of glyceraldehyde 3-phosphate, and remember that now we are in the third phase of glycolysis, which is the energy yielding phase. And also remember that there are always going to be two molecules which will synthesized after every reaction. So glyceraldehyde 3-phosphate is oxidized into 1,3-bis-phosphoglycerate with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. So this dehydrogenase enzyme, they are different, and there is a special characteristic about this enzyme. They are basically oxidoreductases, and these enzymes are important for carrying out oxidation reduction reaction. So let's talk about in this reaction. So here the oxidation reaction involves the oxidation of aldehyde into carboxylic acid, and subsequently there is also a reduction of coenzyme NAD plus to NADH and H plus. So these two are the oxidoreductase reaction, and which is carried out by dehydrogenase enzyme, that is glyceraldehyde 3-phosphate dehydrogenase. Furthermore, the free energy which is produced in the form of inorganic phosphate, this is utilized to synthesize 1,3-bisphosphoglycerate. And this is the first oxidation reduction reaction of glycolysis pathway. Now another important thing about this reaction is there is only limited amount of NAD plus available in the cell. Therefore NADH, which is generated in this reaction, must be reoxidized to NAD plus for glycolysis to continue. So if we don't have enough NADH, glycolysis cannot be continued. So basically there are two different mechanisms through which NAD plus can be obtained by oxidation of NADH. So first is NADH mediated conversion of pyruvate to lactate, and this reaction is only possible when there is not enough oxygen in the cell. That means during anaerobic condition. So during anaerobic condition, pyruvate is converted into lactate, and by doing so, during this reaction, NADH is oxidized to NAD plus, and this NADH then can be used in this glycolysis reaction of converting glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Now the second mechanism is oxidation of NADH via respiratory chain in the mitochondria, and this is possible when there is enough oxygen available. So that therefore this reaction only takes place when during an aerobic condition. So in short, if it is anaerobic glycolysis, NAD plus is obtained by NADH mediated conversion of pyruvate to lactate, and if it is aerobic glycolysis, NAD plus is obtained through respiratory chain mediated oxidation of NADH. So this is something very important to know, that if NADH is not reoxidized to NAD plus, that can actually inhibit the glycolysis pathway. Now the next reaction of glycolysis, where 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. An enzyme which catalyzes this reaction is phosphoglycerate kinase, and as the name suggests, in this case this kinase enzyme transfer the phosphate molecule from 1,3-bisphosphoglycerate to ADP and synthesize two molecules of ATP in this reaction. And this is this type of synthesizing ATP is an example of substrate level phosphorylation, where ATP is formed at the expense of a substrate, and in this case the substrate is glyceraldehyde 3-phosphate. Compared to the other way of synthesizing ATP is synthesizing ATP through electron transport chain in the mitochondria. So remember that this is the first example of substrate level phosphorylation in glycolysis. And this is also the first ATP synthesizing reaction of glycolysis. And another important note of this reaction is, unlike other kinases such as hexokinase, glucokinase, and phosphofructokinase, this reaction is physiologically reversible, because if you remember that the first reaction of hexokinase, glucokinase, and phosphofructokinase, those were irreversible reaction. So this is the only kinase in glycolysis reaction which is reversible. Now in the next reaction, 3-phosphoglycerate is then converted into 2-phosphoglycerate, and enzyme responsible for this reaction is phosphoglycerate mutase. And in this reaction, the phosphate group of 3-phosphoglycerate is moved from carbon 3 position to carbon 2 position. And this is a characteristic of mutase enzyme. So what they do is, mutase are enzymes that catalyze the migration of a functional group. So in this case, the functional group is phosphate. So migration of a functional group within a molecule. So the molecule here, we have the same molecule, phosphoglycerate. The only difference is the phosphate group is moved from a carbon 3 to carbon 2. And this is a special characteristic of mutase enzyme. So this, I'm telling you this because you will remember this reaction very clearly. You wouldn't have to memorize the enzymes for each and every reaction. Now the ninth reaction of glycolysis is dehydration of 2-phosphoglycerate. And as the name suggests, because it's dehydration, so the water is removed from this reaction to synthesize phosphoenol pyruvate, and the enzyme responsible for this reaction is enolase, which produces phosphoenol pyruvate, as well as this reaction also produces high energy enol phosphate of phosphoenol pyruvate. And the last reaction of glycolysis, that is the formation of pyruvate. So in this reaction, phosphoenol pyruvate is converted into pyruvate with the help of enzyme pyruvate kinase. And again, as the name suggests, this kinase transfer the phosphate group from phosphoenol pyruvate to ADP to synthesize 2 molecules of ATP. And this is the second ATP synthesizing reaction of glycolysis, another example of substrate level phosphorylation, and third irreversible reaction of glycolysis. And again, this is also a very heavily regulated enzyme in glycolysis pathway. So now you can see that from one glucose molecule, we now have 2 molecules of pyruvate. And so this pyruvate is the end product of glycolysis. And there are several ways pyruvate can be utilized after it is synthesized. So now let's talk about how pyruvate is being utilized in different ways. So the first condition is aerobic fat condition. So in the presence of oxygen, when there is sufficient glucose level in the body, pyruvate is oxidized into acetyl-CoA, and acetyl-CoA is then further metabolized into mitochondria through TCA cycle to synthesize ATP, carbon dioxide, and water. Furthermore, acetyl-CoA can be also utilized to synthesize fatty acid. And these reactions are important in brain, kidney, sperm cells, as well as any cells with mitochondria, which can use this pathway of glycolysis to synthesize energy in the form of ATP. And the second condition is aerobic fasting condition. So when there is enough oxygen available, but the blood glucose levels are low due to the fasting condition, and that could be due to fasting or starvation or intense exercise. This all can lead to decrease in blood glucose level in the body. So during this time, pyruvate is converted into oxaloacetate, and oxaloacetate is a TCA cycle intermediate, and oxaloacetate is then subsequently converted into glucose through gluconeogenesis pathway in the liver. And you need to remember that during fasting condition, body relies heavily on a gluconeogenesis pathway to synthesize glucose. Why? Because there are some cells and tissues which heavily rely on glucose to synthesize energy in the form of ATP. For instance, brain, red blood cells, kidney, and exercising muscles. So synthesizing glucose through gluconeogenesis pathway during fasting condition is important to make sure that glucose is always available for the cells or tissue to utilize to synthesize energy, and most importantly for brain. Now the third condition is anaerobic condition, where there is deficiency of oxygen. In this condition, pyruvate is converted into lactate, and this pathway can be utilized by exercising muscle, red blood cells which lacks mitochondria, poorly vascularized cornea, as well as during hypoxia, which is common in myocardial infarction, pulmonary embolism, hemorrhage, or during shock. So during this condition, pyruvate is converted into lactate. And here, just to compare that, here we have seen exercising muscle in aerobic fasting condition also and in anaerobic condition also. So for instance, in aerobic fasting condition, it would be just the initiation of exercising, but during intense exercise, when the oxygen level starts to drop, the muscles switch from aerobic glycolysis to anaerobic glycolysis, where it then converts pyruvate into lactate. Now the last condition is in yeast. So during anaerobic condition, so in yeast, pyruvate is converted into acetaldehyde, and subsequently it is then converted into ethanol. And this reaction is the basis for the brewing of beers and the fermentation of grape sugar in making wine. So therefore this reaction is also called as alcohol fermentation in yeast. So these are the different several ways where pyruvate can be utilized to make sure that cells or tissues can use glucose to synthesize energy in the form of ATP. So the last reaction I want to talk about is the reduction of pyruvate to lactate. This is an example of anaerobic glycolysis. So in this reaction, pyruvate is reduced to lactate with the help of enzyme lactate dehydrogenase. And as the name suggests, this is dehydrogenase enzyme. So this is our first example of reduction reaction where pyruvate is reduced to lactate. And the oxidation reaction is a very important one, which I have mentioned earlier, that in this reaction where NADH is reduced to NAD plus. So now this NAD plus is available to be utilized in the previous reaction of glycolysis, that is the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So now you know the importance of NAD plus, because there is not enough NAD plus available, so NADH must reoxidized to NAD plus, and now to make sure that glycolysis pathway is continued. Now I just want to quickly review the glycolysis pathway. So here we have a glucose molecule which is converted to glucose 6-phosphate with the help of enzyme glucokinase, hexokinase, and this is irreversible reaction and at the expense of ATP. Second, glucose 6-phosphate is converted to fructose 6-phosphate. Fructose 6-phosphate is then subsequently converted into fructose 1,6-bisphosphate with the help of enzyme phosphofructokinase 1 at the expense of ATP, and this is the energy investment phase. Furthermore, fructose 1,6-bisphosphate then split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And from here, because only glyceraldehyde 3-phosphate undergoes the third phase of glycolysis reaction and not the dihydroxyacetone phosphate, therefore dihydroxyacetone phosphate must be converted into glyceraldehyde 3-phosphate, and therefore we end up having 2 molecules of glyceraldehyde 3-phosphate. Subsequently, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, where this reaction also generates 2 NADH coenzymes, and subsequently 1,3-bisphosphoglycerate is then converted into 3-phosphoglycerate and produces 2 molecules of ATP, which is an example of substrate level phosphorylation. And furthermore, 3-phosphoglycerate is then converted into 2-phosphoglycerate, and 2-phosphoglycerate into phosphoenol pyruvate, and finally phosphoenol pyruvate is then converted into pyruvate with the help of enzyme pyruvate kinase, another irreversible reaction which also generates two molecules of ATP. And if in case of anaerobic glycolysis, pyruvate is then converted into, is reduced to lactate with the help of lactate dehydrogenase enzyme, which um, which oxidize NADH. And as you can see here that in this reaction we used NAD plus, so NAD plus was reduced to NADH. So here in the last reaction, reoxidation of NADH generates NAD plus, and this NAD plus is now available to be utilized in this reaction for glycolysis to continue. Lastly, I just want to quickly talk about the clinical relevance of this pathway, specifically the condition called lactic acidosis. So here you can see that the one molecule of glucose is broken down into two molecules of pyruvate, and suppose in the presence of oxygen, that is during aerobic condition, pyruvate is oxidized into acetyl-CoA, and subsequently acetyl-CoA is metabolized through TCA cycle in the mitochondria to synthesize ATP, carbon dioxide, and water. And also remember that during aerobic condition, NADH, which is generated through this reaction, reoxidized into NAD plus through respiratory chain within the mitochondria. And this NAD plus, which is reobtained, that can subsequently be used for glycolysis pathway to continue. Now now during oxygen deficiency or during anaerobic condition, this pathway is inhibited, and therefore the NADH, which needs to be reoxidized, cannot be reoxidized through respiratory chain. As a result, the ratio of NADH to NAD plus increases, and when the ratio of NADH to NAD plus increases, the increase in the ratio favors the reduction of pyruvate into lactate. And remember that the only purpose of reduction of pyruvate to lactate is to reobtain NAD plus by oxidizing NADH into NAD plus, and in order for glycolysis to continue. Now also remember that lactate which is synthesized, this is like a dead end of the pathway. So if lactate is not removed, it eventually accumulates in the muscle, causing the intracellular pH in the muscle to go down, resulting in the acidic environment in the muscle and causing muscle cramps. So in order to prevent the lactate accumulation in the muscle, body utilizes this Cori cycle. So during this Cori cycle, lactate is carried from muscle through blood into the liver, and in the liver, lactate is eventually oxidized into pyruvate, and pyruvate is then converted into glucose through gluconeogenesis pathway. And once the glucose is synthesized in the liver, it can then again be transported into blood, and from blood into the muscle, and in the muscle the glucose is reutilized to generate ATP. So remember that a Cori cycle is basically the glucose which is synthesized in the liver from lactate in the skeletal muscle, and then glucose is then reutilized in the muscle to synthesize energy. So this is one mechanism to prevent the accumulation of lactate in the muscle. And lastly, I just want to remember that when the ratio of NADH to NAD plus is high, this favors the reduction of pyruvate into lactate. On the other hand, when the ratio of NADH to NAD plus is low, this favors the oxidation of pyruvate into acetyl-CoA, and acetyl-CoA is then subsequently metabolized through TCA cycle to synthesize ATP, carbon dioxide, and water. So I really hope that you enjoyed this lesson, and most importantly you learned something from this lesson. And if you do so, please like and share the video and subscribe the channel. I'll see you soon next time. Thank you so much.